How do we move these things across the epithelial lining of the GI tract? So we've been speaking about moving things through. We've been speaking about digestion, so how we break them down, from carbs to glucose, etc., and the different juices that are involved in that. Now we're going to look at how we further break them down if needed, and then how we actually move them across the epithelium of the GI tract. Essentially, the mechanism by which we achieve absorption. So our objective here is to describe the fundamental mechanisms involved in the absorption of carbohydrates, proteins, lipids, and their digestion as well, their digestive products. We're also going to explain how the mechanism of lipid absorption is related to the non-polar nature of fats. So when we talked about fats at the very beginning in Unit 1, we would have talked about the fact that they are amphipathic, they are non-polar, they don't play nicely with water for the most part. And so they need a special solution in order to, one, move them around, and then two, move them into our liver. How do we get them absorbed? Because we can't move them into the blood. How do we get them to be absorbed? And we'll talk about that as well. So we'll look at the digestion and absorption of all of our nutrients, including water. We'll talk about carbs, proteins, lipids, and we'll also look at vitamins, minerals, and then at the very end, how we absorb water. Now, it's going to call on us to remember some of these transport mechanisms. So at the top, we're looking at one of the main mechanisms that helps with this. Can anyone identify what's happening at the top? What is the top, this entire panel here representing? So the sodium potassium pump I'm seeing here in the chat, which is correct. Someone is also saying facilitated diffusion, but that is the bottom panel, right? But the top panel is definitely the sodium potassium pump, okay? And so these two mechanisms we would have talked about before, initially in Unit 1, and then we would have also just recently reviewed them as a part of looking at the kidneys and how reabsorption takes place in the kidneys. And so a lot of the same mechanisms that we saw for the movement of certain particles across the tubular structures of the kidney also apply here for the movement of some of our micronutrients across our epithelia of the GI. So just calling on us to remember and recall the sodium potassium pump on the basolateral surface of most of our cells, including our GI tract cells, and then the facilitated diffusion being the means by which we can get some of our nutrients into the cell using the energy of the sodium because of the gradient that is set up by the sodium potassium pump. Let's start with carbohydrates. These are usually the bulk of our diet. Carbohydrate digestion, we mentioned, starts in the mouth, and carbs must be broken down to monosaccharides. So they must be broken down to their smallest monomer units in order to be absorbed across our GI epithelial cells. So when we think about disaccharides, polysaccharides, they must be further broken down. Starch begins in the mouth, and when we have the action of salivary amylase and even pancreatic amylase, that sometimes breaks more complex starches down to disaccharides or limit dextrins. And limit dextrins are just these branched polysaccharides, so branched chains that end up at the end product of some of our carbs being broken down, similar to disaccharides. And so salivary or pancreatic amylase will bring it down to these two components, but we must break it down further to those monosaccharide monomers in order for them to be moved into the bloodstream, okay? And so when we have things like our disaccharides, so for example, maltose, sucrose, lactose, those must be broken down into monosaccharides. Can anyone recall what our three common monosaccharide examples are? So glucose is one, it's already on the slide. Anyone can remember at least one or two of the others? So not sucrose. Sucrose is going to be a disaccharide. Going to be glucose and fructose. Oh, there, I gave it away. So galactose is one I'm seeing in the chat here, galactose and then fructose, right? So our monosaccharides are going to be glucose, galactose, and fructose. So those three options can be absorbed across our epithelial cell and go immediately into the bloodstream. Okay. So here's what that absorption would look like. Again, the mechanisms here should look very familiar. We also want to remind ourselves of the nature of this cell. So this is going to be one of our intestinal epithelial cells. It's going to have that microvilli. So it's going to be one, it's going to be on a villus. So it's going to be on the outpouching that we would have described earlier. And then if we look at the cell itself, the cell is also going to have the apical modifications that are called microvilli. Microvilli. And these microvilli are forming what we would have termed as the brush border, really extending that surface area and allowing for more space for absorption to take place. Now, across the apical surface of our epithelial cell, we're going to see sodium moving in passively. Glucose, which is the monomer, an example of the monomer, but fructose can also be using this mechanism. Galactose can also be using this mechanism. It's pretty much going to hitch a ride via that facilitated diffusion that we would have described earlier. So because of the sodium-potassium pump churning away at the basolateral surface, creating the gradient for sodium to come in passively, we can take that energy and bring in some of our monosaccharides, glucose, galactose, fructose being the examples. And that is how we get them inside the cell. From the inside of the cell, they can move across the basolateral surface. So now they're at a higher concentration inside the cell, a lower concentration in our underlying capillaries, and then we can absorb them right into the underlying capillary bed via facilitated diffusion. Okay, so this should be secondary active transport. And you can definitely make a note here. Actually, let's see if I can make a note here so that I capture this. So this mechanism we said is secondary active transport. Okay. Down here, we have the sodium-potassium pump, and then this mechanism is facilitated diffusion. And so this is why it's so important to have the underlying capillaries in association with the epithelial layer. So just kind of relating this to what we spoke about in terms of the layers of the GI. So we've got the epithelial layer, right? That is the innermost part of the mucosa. And so having that lamina propria, which is the underlying connective tissue underneath the epithelial layer, is how we can facilitate that absorption into the bloodstream. The small capillaries that are found in the lamina propria help to facilitate that absorption into the blood. Alrighty. Now, proteins are broken down by proteases, and there are several different types of proteases. So proteins can be broken down by endopeptidases. Endopeptidases are going to split the peptide bonds toward the interior of that peptide chain, whereas exopeptidases are going to split or cleave the external fragments of that polypeptide bond or polypeptide chain. So essentially, endopeptidases will cut anywhere within the center of that peptide chain. Exopeptidases will cut anywhere towards the periphery. So essentially cutting the amino acids off at the end. And then the third class of proteases are zymogens. And zymogens are essentially these granules that are stored. They are the inactive form of proteases, and they're going to be secreted by exocytosis and then activated by proteolysis. And I'll talk about that coming up on the next slide. But essentially, several different inactivated proteases are stored in these granules. And if you were to take the pancreas, cut it in apart, do a frozen section, look at it under the microscope, you would actually see these granules with all of these inactivated proteases stored inside. So inside those granules, we've got all of these inactivated proteases that are not quite active. I'll talk about how they become active on the next slide, but then we want to talk about where proteins breakdown begins. So we know protein digestion starts in the stomach. We would have talked about this yesterday. It's under the influence of the enzyme pepsin. And pepsin is secreted in its inactivated form as pepsinogen, which is secreted from the chief cells. If you remember us speaking yesterday about the cells of the stomach, the chief cells, right? Some of these gastric pit cells, chief cells being the ones that secrete pepsinogen. Pepsinogen was activated to pepsin under the influence of the acid that is found in the stomach. So when they release this pepsinogen out into the stomach lining, acid, which is secreted from the parietal cells, is what activates that pepsinogen to its active form, pepsin, and then it can break down proteins to their smaller peptide fragments, okay? So essentially, both of these cells are playing a role in the activating of pepsinogen to pepsin, and then that pepsin can begin breaking down our proteins in the stomach. Now, getting to these zymogen granules, right? So the zymogen granules that I was just talking about in the pancreas has all of these inactivated proteases within it. It's got trypsinogen, it's got chymotrypsinogen, it's got procarboxypeptidase. And we're essentially going to activate these when they get into the lumen of the intestine. This is why the pancreas does not digest itself, because these are being stored in their inactivated form, and they won't be activated until they're actually released into the ducts and make their way to the small intestine. So what starts this activation off is the cells of the small intestine lining itself. These enterokinase cells are going to activate one of our proteases. So trypsinogen only is activated by enterokinase to the active form trypsin, okay? From there, trypsin will then activate the other two proteases. So trypsin will activate chymotrypsinogen to chymotrypsin. Trypsin will also activate procarboxypeptidase to carboxypeptidase. And then we have this cocktail of several different proteases that can kind of just bombard what's left of our undigested protein coming down from the stomach and complete that digestive process here in the small intestines. Okay, so it's really important that the brush border proteases, which are enterokinase and aminopeptidase, activate trypsinogen, which is the only one that they activate. So they only activate one of these, and then trypsin will kind of take the job, take over from there and activate the others.